Greetings to all who have joined with us today. I'm Hemant Sistla, working as a scientific associate at thinkMolecular Technologies Private Limited, Bengaluru, India. thinkMolecular is a molecular simulations company with its focus in drug discovery. At the very outset, I would like to thank our founder, Dr. Ramesh Sistla, for his invaluable inputs in this work. Today, I'll be talking to you all about the RNF114-BRD4-PROTAC ternary complex modeling. In 2019, Spraden et al. Identified a naturally occurring compound, nimbolide, as a ligand of the ligase RNF114. In this study, they demonstrated the degradation of the bromodomain protein BRD4 using nimbolide, which was incorporated in a PROTAC. Now in 2021, Luo et al. From the same group identified an analog of nimbolide EN219 using chemoproteomic approach, and this was also deployed in a PROTAC to target the same protein. This is the structure of the PROTAC developed by Luo et al. This is the EN219, which binds to the E3 ligase, and this is the warhead of the PROTAC, which binds to the target. Now, for the rest of my presentation, I'll be referring to this PROTAC as compound 1. Now, the objective of my work was to create a ternary complex model of RNF114-BRD4 and compound 1 using protein-ligand docking, protein-protein docking, and molecular dynamics simulations. Now, RNF114 is a 228 amino acid E3 ubiquitin ligase with a mass of about 26 kilodalton. Now, the thing is that a 3D experimental structure of RNF114 is not available. Now, this here is the image of the blast output of RNF114. So this bar here indicates the sequence identity of these various proteins with that of RNF114. Now, as you can see, the sequence identity of many of these proteins with RNF114 is quite decent. But then the sequence overlap is poor. And therefore, these proteins cannot be used for building a homology model of RNF114. Now, even the best among these results, RNF125, whose domain diagram is shown here with RNF114, even this cannot be used for building a homology model for RNF114 because the available structure of RNF125 does not have the entire sequence overlap with the does not provide the entire sequence coverage with respect to the RNF114. Now, since an experimental structure nor homology model of RNF114 is accessible, we chose to utilize the structure reported in AlphaFold. So RNF114 is a zinc binding ligase. That is, it has 5 zinc fingers, of which 2 are present in the ring domain itself. Now, zinc atoms were incorporated into this AlphaFold structure by taking the structure of RNF125 as a template. The figure on the left shows the AlphaFold structure. Now, this region here, the N-terminal region marked in orange, indicates that the confidence of the model for this particular region is quite poor. Now, according to the literature, this is the cysteine where the EN219 and the nimbolide get covalently attached. But this region does not form a pocket where the ligand can make meaningful interactions. And therefore, we had to subject our RNF114 to a 100 nanosecond molecular dynamic simulation so that the terminal regions, the N-terminal and the C-terminal regions, can fold and make the structure compact so that the ligand can have meaningful interactions. Now, here on the right, you can see the trajectory of the 100 nanosecond molecular dynamic simulation that was run using GROMACS. This trajectory was then clustered, and the center of the largest cluster was taken forward. So this is the chosen structure of RNF114. As you can see here, this is the pocket where EN219 is supposed to bind, and this is a cysteine 8 where it gets covalently attached. Now, RNF114 was used as the receptor onto which EN219 was covalently docked. Now, this was done using the software suite ICM, which is provided by MolSoft. So this figure here shows the key interactions made by the EN219 on our protein. Now, the regions in green represent the hydrophobic regions, while those in gray are the ones that are exposed to the solvent. So as you can see, this part of the EN219, the bromophenyl, is buried into the hydrophobic pocket, and the other bromophenyl is exposed into the solvent. Now, this is where the linker is attached for a PROTAC to be constructed. From our studies, we also concluded that the R isomer of EN219 is likely to be active on RNF114. Now, allow me to refresh your memory of our compound 1. So this is the EN219, which binds to the E3 ligase RNF114. And this is the warhead JQ1, which binds to the bromodomain protein BRD4. So please bear with me as I explain to you the significance of this crowded picture. So to generate the binary complex, the X-ray structure of JQ1 bound to BRD4 was docked onto the arrived structure of RNF114. So this is the structure of the RNF114. It was treated as a receptor onto which the BRD4, the BET1 domain of BRD4 was docked. So this was treated as a ligand. So during this process, about 10,000 binding modes were generated. Now this was done using the protein-protein docking module of ICM. So as 10,000 modules, different binding modes were generated. Some of those modes are represented here. So these 10,000 binding modes were then clustered basing on the interaction fingerprints. So these interaction fingerprints are nothing but a representative of the different patterns in the interaction between the bromodomain protein and the RNF114. So the regions here, you can see the different colors here. All of these represent the different clusters. So what we did was we took clusters from these clusters, and then basing on the docking score, the interaction energy, we shortlisted these cluster centers. And then we got a couple of binary complexes. Now, these were again shortlisted based on the protactability of these individual ligands. So these ligands here, so what I mean by protactability is that basing on how close these ligands are, what are their orientations, whether they can make the PROTAC which we have designed. So basing on these considerations, we have shortlisted them further to about 50 binary complexes. Now, these potential binary complexes were then further subjected to energy minimization and short ND runs using OpenMM. So the binary complexes arrived thus were then subjected to this PROTAC docking. So that is our compound 1 was covalently docked onto this binary complex RNF114 and BRD4. So the docking modes of the PROTAC were assessed based on the key interactions that the individual ligands EN219 and JQ1 made with the respective proteins. So as you can see in this picture, this is the compound 1, and this is the EN219 part of the PROTAC, and this is the JQ1 part of the PROTAC. So this binds to our E3 ligase RNF114, and this is the part which binds to the BET1 domain of BRD4. And this is the linker which joins the two ligands and completes our PROTAC. The figure on the right indicates the interaction diagram. That is, it indicates the key interactions which are PROTAC, which are compound 1, makes with the binary complex. Now, the regions here, these regions in the green, as I mentioned earlier, they indicate the hydrophobic regions, while those in the gray are the ones which are exposed to the solvent. Now, to summarize what the work which I have done, so this workflow succinctly summarizes the efforts taken up during this work. Now, in which a functional version of RNF114 was built. Then the active chiral conformation of EN219 was identified, which was then docked onto the RNF114 structure. Then a binary complex of RNF114-BRD4 was obtained using protein-protein docking. Subsequently, the compound 1 was docked onto this binary complex to make the ternary complex, which was the objective of our work. Now, in this process, protein ligand docking, protein-protein docking, molecular dynamics, and trajectory analysis were the key techniques and tools that were employed. So we look forward to putting the ternary complex in perspective by introducing ubiquitin-bound E2 ligase into the picture. So this way, we hope to gain structural understanding of the feasibility of ubiquitination of the target. We are also very keen on using our workflow on building more ternary complex models and gain useful structural understanding for the prospective designs of the PROTACs. Now, this kind of work is being keenly pursued at thinkMolecular. So as you can see here, this is the androgen receptor, and this is the E3 ligase cereblon. And this PROTAC here is ARV110. So ARV110 is a clinical candidate which is currently in phase 2 clinical trials, and this has been designed by Arvinas, and this is an androgen receptor degrader. So this is a ternary complex which we have built, which we have modeled, and we are quite sure of its. We are quite confident in its structure, and we do hope to compare this with the experimental structure of this ternary complex whenever it is available. Now, I thank Dana-Farber Cancer Institute for giving me this opportunity, and I thank each one of you for your very patient listening. Now, you can write to us at info@thinkmolecular.in for the coordinates or any other queries that you might have. Thank you again for your patient listening.